Okay, so first thing first, we need 3 lessons, right? So everybody remembers that. The first couple of slides that you see here on the one that I sent you, the PowerPoint, we aren't going to be using them today. We're going to be using them on our 5th lesson in histology, because the first slides are talking about development of the blood. How can I make blood? It's called hematopoiesis, right? So production of blood. Hematopoiesis will be asked in histology self-control test. So even though it is an embryological topic, they won't ask you in embryo self-control test about it, but in histo. So as a result, we'll talk about it in histo. So unfortunately, or fortunately, you're going to be skipping all the way until this slide, okay? Because that's more relevant for us right now. So first thing first, we're going to start talking about the gametogenesis. Okay, gametogenesis is the production of gametes. Gametes, as you know, are the sex cells. We have 2 of them. We have the oocytes in the female, and we have the spermatozoa in the man. So these are the gametes. These are the sex cells in the human body. But before we start talking about the gametogenesis, we need to look at this picture a little bit. I think you know that your recommended book is the Langman Embryology. This is a very horrible book. It has lots and lots of mistakes. Anatomy department is aware of those mistakes, so they tend to test you on that. Okay, so in the lectures, they will typically tell you that this is a mistake in the book. Okay, so if you attended the lecture, you would know. But if you didn't, if you just read the book, they will catch you on that. So these pictures that you see here, they are from the book, from the Langman Embryology. Don't buy it, you don't need it. The only thing you need is definitely the slides, me, and past papers. Okay, so that's the only thing you need. So this embryo that you see on this picture is actually 4-week-old embryo. So it's 4 weeks, it's 1 month exactly. What does this embryo consist of? Well, you can see that it has a head, it has a heart. The heart of the embryo is actually in its neck, because that's how young it is. It's just 4 weeks old. So we can actually see that the heart is in the neck of the embryo. It has the back. It has the anterior abdominal wall, but you can't see it. You cannot see the belly of the embryo. Instead, you can see that something is attached to the surface of the belly like a blister, right? Imagine that here on your anterior abdominal wall, you have some kind of blister just sitting here, right? This blister, this sac, this is known as the yolk sac. It's a very important structure in embryo. We'll talk about it. It arises on day 14 during the 2nd week, and it disappears during the 3rd month of the development. Okay, so it's not there for long, because if you look at your anterior body wall right now, you can see that there isn't any yolk sac present, right? So this is the back. This is the body wall of the back, right? And this is anterior body wall, and this is the yolk sac blister. If this is the head, that means that on the opposite end, we're going to see the tail. Okay, so this is where the tail will be present. Or in simple English, that's the place for the ass. Right here, something is attaching to your embryo, to this area. This is your future umbilical cord. And you remember in histo we talked about it, right? We had a slide for umbilical cord. It was containing the blood vessels, which were going from the embryo to placenta, and from placenta to the embryo. So you can see that umbilical cord is trying to attach to the embryo right now. That means that this area right here, this is your future belly button. So somewhere here, that's where the belly button will be. We can see that your embryo is sitting in some kind of sac as well, this cavity where the embryo is. This cavity is known as the amniotic cavity. This amniotic cavity is filled with the amniotic fluid. And for sure, in movies, you have heard this thing before, like they say, my water broke, right? In the movies, they keep screaming, my water broke. What does it mean? It means that this amniotic cavity has ruptured. So all of the amniotic fluid is now on the floor. The baby is ready to be born. Okay, so that's it. And it's funny because in movies, they always show that, I don't know, 20 minutes after the water broke, she gives birth. In real life, that's not the case. It takes 12 to 24 hours after the water broke for the baby to arrive. Just FYI. Okay, so movies are definitely false representation. Yeah.
I have a question. It might be stupid, but when you say the embryo, do you mean like everything including the amniotic cavity or just
No, just the embryo itself. Okay, so embryo is just this little tiny structure in the middle, right? So this area in the middle, which is technically between your amniotic cavity and yolk sac. So that's embryo. So you say that embryo is located in the amniotic cavity. It's actually its home for 9 months, right? So this embryo is 4 weeks old. We're starting to talk about gametogenesis. We said that the gametes are oocyte and spermatozoa. Are they haploid or diploid?
Diploid.
They're haploid. Oocytes and spermatozoa are sex cells and they're haploid sex cells. What does it mean, haploid?
Single set of chromosomes.
Exactly. It means that they possess only one set of chromosomes. So if you look at the oocytes, if you look at 100% of oocytes, each and every oocyte inside will have only one chromosome, which is X chromosome. So I repeat, total amount of oocytes, 100% of them, every single oocyte on the planet has one chromosome, and that chromosome is X chromosome. But spermatozoas are a bit different, because if you look at one man and you look at the spermatozoas of that man, half of the spermatozoas, so 50%, will actually have X chromosome. But other half, another 50%, will have Y chromosome. So in females, oocytes, all of them have one chromosome, hence they're haploid, and that one chromosome is X chromosome. In males, the total population of spermatozoa are divided. Half of them have X chromosome, half of them have Y chromosome. So sex of the embryo, gender of the embryo, or human, doesn't matter. What does it depend on? What does the gender depend on?
the spermatozoa?
For sure, it depends on the spermatozoa. Because if we have a spermatozoa carrying X chromosome and it fertilizes your oocyte, we have a diploid structure called zygote, which consists of XX, right? If we fuse the oocyte with the spermatozoa carrying X, we have a fertilized oocyte called zygote. It will contain XX chromosomes. So this is your future girl. But if we have a spermatozoa carrying Y chromosome, right, it will fuse with the oocyte carrying X, you're gonna have a boy. So sex of the human, sex of the baby, it definitely depends on the spermatozoa. It depends which exact chromosome is it carrying. Is it carrying X or is it carrying Y? Is it clear until this point? The problem with embryo course is that you constantly have to ask yourself this question. You have to ask yourself, where does it come from? Okay, you look at the oocyte, you say, where does it come from? Immediately. Next semester oral, that's the entire embryo for you. Just one question you will hear constantly, where does it come from? Another question that you could hear is, what does it give rise to? It's just the opposite way, yeah? So where does it come from? Where does it give rise to? We're starting to talk about gametogenesis for the first like 15 minutes, right? We still can't start. Gametogenesis means production of gametes, right? Formation of oocytes and spermatozoas. So we have to ask ourselves, where do oocytes and spermatozoas come from? Both of these cells come from these cells. Oocytes arise from PGC. I will be constantly saying PGC. PGC means primordial germ cell. Okay, so oocytes, they come from the PGCs, but also spermatozoa. Both oocytes and spermatozoas arise from the primordial germ cells. So how do you know if you're making an oocyte or if you're making a spermatozoa, right? So how do I know that this particular PGC has to give rise to the oocyte, right? How do I know? We said that the sex cells are haploid, but PGCs are actually diploid. If you look at a PGC and it is diploid, it could be either carrying XX or it could be carrying XY. Diploid means that it has two sets of chromosomes. These two sets could be either XX or XY. If we have a PGC carrying XX chromosomes, later it will be dividing, it will be splitting and giving rise to two cells. Each of these cells now has one X chromosome. How do you call these cells?
The oocytes themselves?
They're oocytes themselves. Because all of them, I know there are just two of them, but all of them have one X chromosome. So how do I know if PGC will give rise to the oocyte? It will give rise to the oocytes if PGC has XX chromosome in itself, right? If PGC carries XX chromosome, it will give rise to the oocytes. Take a look at this PGC. It has XY chromosome. The same idea. This PGC will be splitting, right? So it will be dividing, it will be splitting. It will give rise to two cells now as well. The problem is that one cell will have X chromosome. Another cell will have Y chromosome. So this population is now divided because half of this population has X chromosome, half of it has Y chromosome. Where do we describe something like that? Of course, for spermatozoa. Do you get it? PGC carries XX, is a diploid cell. When it divides, it gives you two haploid cells. Each of this haploid cell has X chromosome. These cells must be oocytes because 100% of them have X chromosome only. This PGC has XY chromosome. So when it splits, it also gives rise to two haploid cells. The problem is that half of these haploid cells have X and half of these haploid cells have Y. Can it be oocytes? Of course not, it can't. It has to be spermatozoa because we just said that for spermatozoas that's the case, that half of them have X, half of them have Y. Is that clear?
Probably getting ahead of myself, but like, how is the X population not oocytes? Like,
how can you have one cell give rise to oocyte and spermatozoa? How is it possible?
That's what I'm asking.
If it's possible, then you have like, I don't know, like, can you imagine one cell can give rise to two immediately, a boy, like an oocyte and a spermatozoa? It's impossible.
Yeah, that's what I'm asking. So the process
we don't
is
look
different.
at them individually. We don't look at them individually. We look at them as a population, as a whole.
Okay.
So as a whole population of these cells, all of them have X chromosomes, right? On this picture, all of them have one X chromosomes. They must be oocytes because what did I tell you? only oocytes, all of them have one X chromosome. In this population, can you have a cell that has Y chromosome?
No, this is what I got. I just like
Yeah.
process of like dividing, it's different for each, right? Like when it divides from the diploid to the haploid.
Yes, so when it divides, you see that one diploid cell, PGC, gives rise to two haploid cells because they have only one X chromosome here, right?
Okay.
Yeah. For PGC carrying XY, it's the same idea. The only difference is that there is Y. If there is Y, for sure, these are spermatozoas. So I repeat it. How do you know if PGC will give rise to the oocytes? This PGC has to have XX chromosomes. So when it splits, it gives you two cells, X, X. How do you know if PGC is going to give rise to the spermatozoas? If it has XY chromosome. So sex cells are haploid, PGCs are diploid. Is it clear?
And just one question to be sure. So PGC, they can be both XX and XY, or?
In the same human being, no.
Mm-hmm.
In the same human, they cannot. You cannot have a human which is both a male and a female, can you?
No.
No. So this is why in one human, you could have PGCs which have only XX. In another human, you could have PGCs which are only XY.
Mm-hmm.
What does it depend on? It depends on the moment of fertilization. When you started making an embryo, did you make a boy or did you make a girl at the genetic level, right?
Thank you.
Uh-huh. I see Noam is not happy. What's up, Noam? What are you getting not?
No, I'm typing everything you're saying.
Okay. Because your face looks like you are not.
I'm just concentrating.
Okay, good. Okay, so this is the situation. So we understood that we have the gametes, oocyte and spermatozoa. They come from PGCs. We have different types of PGCs depending whether you're a boy or a girl, right? So as a girl, when I was an embryo, my PGC only were containing X and X, right? As a boy, X and Y. Where do PGCs come from? That's the problem with embryo. Now we have to ask where they come from. How old was this embryo? As you remember, 4 weeks old. Now we have to look at an embryo which is 2 weeks old, just 2 weeks younger than that. How do we call the top cavity?
Amniotic sac.
Very good. We call it amniotic cavity. Always, the top is amniotic cavity, always. How do we call the bottom cavity?
The yolk sac.
The yolk sac. Always, it is the yolk sac. What should be here in the middle?
Embryo.
The embryo itself. Okay, so this is what should be there between the two cavities, between the amniotic and the yolk sac. That's where the embryo should be. But we're drawing a picture of a 2-week-old embryo, 2 weeks. At this moment, the mom realizes that she's pregnant, okay, more or less. So she's starting to experience that there is like 1 or 2 days delay in the menstruation, right? So she isn't getting the menstruation. So she isn't aware yet fully, but she's starting to suspect it. So can I have a full embryo here? Not really. When you have a 2 weeks old structure, this 2 weeks old structure actually consists of 2 layers of cells. That's it. Your embryo, if I may, during the 2 weeks old time, it consists of 2 layers of cells. That's it. The top layer is known as the epiblast. The bottom layer is known as the hypoblast. Together, these 2 layers of cells have a name. They're called bilaminar germ disc. Okay, bilaminar germ disc. So I really want you to see how fast things are working in embryo, right? 2 weeks old embryo, there's nothing. There's just 2 layers of cells. Epiblast being top, hypoblast being bottom. That's it, just 2 layers of cells. Literally 2 weeks later, you look at the 4-week-old embryo, what do you see? You see already a head with the brain, heart, back, front, ass, all of the structures are arising in just 2 weeks. So things are working very, very fast in embryo. We're trying to answer the question, where do PGCs come from? PGCs are arising from the epiblast during the 2nd week. Okay, so this is where they come from. They come from the epiblast during the 2nd week. So let's see if you get it correctly. How do I call, which kind of gametes do you know?
Oocyte and spermatozoa.
Are they haploid or diploid?
They're haploid.
Which chromosomes are in the oocyte?
XX.
But they're haploid. If you say X,
Oh, it's just the
it means diploid. But oocytes are haploid, so they can only have
X.
just the X, just the single X. That's what makes them haploid. So every single oocyte, doesn't matter how many there are of them, inside of them, inside of the nucleus of the oocyte, there's one X chromosome, that's it. Spermatozoas are also haploid, but
they're either X or Y.
exactly. Let's imagine in a man, for example, I have 100 spermatozoas, 100 of them. How many will have X chromosome?
About 50.
About 50. How many will have Y chromosome? About 50. Do you get it? So in spermatozoas, we don't have only X, because if we had, we will only have girls on the planet. That's it. Right? So half of the spermatozoas have X, half have Y. Where do gametes come from?
The PGC.
Are they haploid or diploid PGCs?
Diploid.
They're diploid. If I want my PGC to give rise to oocytes, which chromosomes should it contain?
XX.
XX. If I want my PGC to give rise to spermatozoas, it should contain
XY.
XY. Where do PGCs come from?
The epiblast
Epiblast during
in a 2-week embryo.
During 2nd week. Okay, so in embryo we just say 2nd week of embryological development, but it's mouthful, so just say 2nd week. Now, this is not the area where PGCs should give rise, right, to things they're meant to give rise to. The area where PGCs will become oocytes or spermatozoas is actually your testes or ovaries, right? So if PGC has XX, is meant to become oocytes, this place where it happens will be ovary. The same for spermatozoas, but in the testes. In embryo, we don't call it testes, we don't call it ovaries. It has a name and it is called gonads. Okay, so in the embryo, testes or ovaries are called gonads. So these PGCs, even though they arise from the epiblast, the area where they need to go to do their thing, to become gametes, is actually gonads. So how does it happen? As soon as the PGCs arise from the epiblast, they detach and they start to migrate around the wall of the yolk sac, reaching the posterior wall of the yolk sac by the 4th week. We can see migrating PGCs at the posterior wall of the yolk sac. Stupid question, how do you know that this is posterior wall of the yolk sac? Oh, actually, apparently it's not stupid. Okay, so how do you know this is a posterior wall of the yolk sac?
Because there's the tail.
Exactly, because that's where the tail. On the opposite end, we have the head. That means that this would be the anterior wall of the yolk sac. Okay, so this is the anterior wall. So I repeat, 2nd week, PGCs arise from the epiblast, detach, migrate around the wall of the yolk sac, reach the posterior wall of the yolk sac during the 4th week. Why are they detaching? Where are they going? They're going towards the gonads. Gonads are paired structures which are located in the lumbar region of the embryonic back. So somewhere on the lumbar side of the embryonic back, you will see your gonads. So PGCs are reaching the gonads during 5th week. 3 numbers are important for PGCs. 2nd week, they arise from epiblast. 4th week, they are at the posterior wall of the yolk sac. 5th week, they are reaching the gonads. Is it clear? There is something called polar bodies, and it's very important. They're going to ask you a question about it. It's not the same as in the genetics polar body. It's a bit different, yeah? So without going too much into the details, take a look at the spermatogenesis and oogenesis. In men, you're going to see that every time a parent cell divides, it gives you 2 daughter cells, right? I think it's a very basic piece of information. Parent cell divides, 2 daughter cells. In men, these daughter cells are all nice and functional. Everything is great. In female, there's a bit of a problem because every time the oocyte divides, it gives rise to the normal oocyte and to the polar body. So what is a polar body? It is the oocyte with little to no cytoplasm. Every time oocytes are going to be dividing in the female, proliferating, they give rise to the normal oocyte and to the polar body. I repeat, polar body is oocyte with little to no cytoplasm. So take a look, oocyte divides, it gives you normal oocyte and polar body. The question is, why is polar body dividing? Because do you see, it also gives rise to 2 cells, which of course are going to be polar bodies as well. But how is it still capable of dividing? Why is it still dividing the polar body? We said it has little to no cytoplasm, but what does it have to divide?
Chromosome?
The nucleus, yeah, the chromosomes, the nucleus, and etc. Okay, so in the self-control test, and especially next semester exam, you're going to see that they will make this very interesting question. They will say that polar bodies are inactive. That's not true. Okay, so polar bodies aren't inactive. They're capable of division. That means that they are active. We said that polar body has little to no cytoplasm. So where did all of its cytoplasm go? To the other one. How do we call division of the cytoplasm in a cell?
Cytokinesis?
Very good, we call it cytokinesis. Is cytokinesis equal in oogenesis? No, because you see that one cell gets more of the cytoplasm than another. So this polar body, I repeat, is the cytoplasm with little to no cytoplasm because of unequal cytokinesis. So in oogenesis, you have 1 parent cell, you have 4 daughter cells, the same as in the male situation, but out of these 4 daughter cells, only 1 of them is functional. Okay, only 1 of them is functional. Only 1 of them can get fertilized. Polar bodies cannot be fertilized because little cytoplasm is not good. As a cell, you still need a bit of a cytoplasm. Clear? Let's talk about spermatogenesis. Okay, so everything I'm going to be talking about here is written here and somewhere else. Okay, so let's talk about spermatogenesis. Production of spermatozoa. So where do spermatozoas come from?
PGCs.
And these PGCs have which chromosomes?
XY.
Yes. So these PGCs, they have to carry XY chromosomes. These PGCs carrying XY chromosomes are at the border of the gonads. That means that this is 5th week, because you remember I just told you 3 numbers are important for PGCs, 2nd, 4th, and 5th week. So during the 5th week of embryological development, we can see that these PGCs are ready to go into the gonads. As you remember, gonads are undifferentiated sex organs, which are destined to become testes or ovaries, right? So basically, to simplify, gonads are your testes or ovaries. What gonads consist of in embryo? Well, there is surface cuboidal epithelium. Okay, so on the surface of the gonad, you will see simple cuboidal epithelium, surface cuboidal epithelium. But the core of the gonads will consist of the connective tissue. Don't forget we're in embryo, so how do we call embryonic connective tissue?
Mesenchyme.
Mesenchyme. So in the core of the gonads, you will find mesenchyme. What is so special about mesenchyme?
Pluripotential.
Very good. It possesses pluripotential stem cells. Please remember that because it's important. So here I have my gonads. These gonads consist of the cuboidal epithelium and mesenchyme, right? Here I have my PGCs carrying XY chromosome, 5th week. What's happening? 5th week, these PGCs are starting to invade the gonads. And as they invade the gonads, they start to divide here. The question is, as these PGCs come into the gonad, do they divide mitotically or meiotically? What do you think?
Mitotically.
Or meiotically? I hear meiotically and I hear mitotically. Actually, it is mitotically. Why mitotically? Because they want to make as many replicas of themselves as possible. The amount of PGCs which is coming to the gonad is very, very little. So they want to make as many copies of themselves as possible to fill up the space in the gonad itself. So 5th week, PGCs carrying XY chromosome come into the gonad and divide mitotically. As they divide mitotically here, they don't just assume a random position. It's not like one PGC is here, another PGC is there. No. These PGCs start to align themselves into a very nice structures. Okay, they start to align themselves into very nice areas. These areas are called cords. So on my picture, what you see here, every dot, every little dot is a PGC. And do you see that these PGCs are starting to align into cords? Next semester, this is Professor Antol's favorite question to ask. What is the difference between cord and a tubule? What is the difference between cord and a tubule?
Cord is aligned cells, and tubules are like intercellular or organelles.
Say better, just tubule, they have something that cords do not.
Openings?
You could say opening, okay, or like a lumen, okay? They are hollow. Tubules are hollow. They have a space inside. They have a lumen. Cords do not have a lumen. They're filled with something. Like look at my, I don't know, this cord right here, right? So from my, I don't know, hard drive. So this cord, if I cut it right now, it is filled with wires. It doesn't have a lumen. It's a cord, right? Look at my pen right here. It has a lumen of some sort. So technically a pen is a tubule. It's not a cord, it's a tubule. So that's the major difference. And you won't believe, but actually, like I personally know 3 people who failed C-chance because of that. Okay, because of this question, what's the difference between cord and a tubule? But you know it now. Okay, so that's the difference. Cords do not have a lumen. Tubules do have a lumen. So these structures right here, these are your sex cords. That's their name. So these sex cords, I repeat, are consisting of a bunch of PGCs. Which chromosomes these PGCs have? They have X and Y chromosome. I repeat it again. 5th week, PGCs, XY chromosome, come to the gonad, divide mitotically, make as many replicas of themselves as possible, align themselves into cord. So these sex cords consisting of PGCs. Let's compare the situation with a female. If I had here a PGC carrying XX, right? It's meant to become oocyte. So this process happens in the female. So this PGC will do the same thing. It will also come into the gonads, also divide mitotically, also align themselves into the sex cords. But in the female, sex cords do not persist, while in the male, they do. So why? Okay, so that should ask you, that they would ask you like, why? Why sex cords persist in the male and in the female, they don't? This is Professor Antle and Professor Rosa's favorite question, especially next semester, especially if they see that you're very stressed, very nervous. If you get this topic, they're gonna be like, why are you a female? Why are you a female? Okay, and it's not like in a nice form, yeah? It's like, why are you a female? It's like, sorry, no? So let me know, why are you a female? For sure you know the answer, guys, so come on. Talk a little bit so my voice can rest.
Because I have XX chromosomes.
You and 95% of other females will say the same thing, including me in the first year. That's exactly what I would say, that I'm a female because I have XX chromosome. But actually, that's a wrong way to answer. It's a bit sexist, but actually I'm a female because I don't have a Y chromosome. Okay, that's a proper way to answer. I'm a female because I don't have a Y chromosome. What is so special of the Y chromosome? This is the long arm of the Y chromosome. This is the short arm of the Y chromosome. On the short arm, in the corner, in the end of the Y chromosome of the short arm, there is a gene here. This gene is known as the SRY gene, or in our name, sex determining region of the Y chromosome. This sex determining region of the Y chromosome is coding for a factor. This factor is known as the testis determining factor. For PGCs to stay in the cord, to persist as the cord, TDF is needed. So once PGCs come into the gonads, once they divide here mitotically, once they align themselves into the sex cords, they check. Do we have TDF? We do. We stay as the sex cords. In the female, PGCs also align into the sex cords. But is there a TDF in the female? No. Why not? Because there is no SRY, because there is no Y chromosome. So as a result, in female, PGCs dissociate into clusters. They don't stay as the cords, they simply become clusters. Is this part clear? So quickly, how do you call this? What is this?
Cord.
A cord.
It's a sex cord, and the sex cord is consisting of
PGCs.
PGCs. If this sex cord persists, these PGCs in it have
Y chromosomes.
XY, because they're diploid, right? So PGCs are diploid, they have XY. But if this sex cord dissociates into clusters, that means that these PGCs actually have XX chromosome. Okay, this is 5th week. You're gonna see that simultaneously as we're making the sex cord, some surface epithelial cells detach and migrate into the gonad and sit there. Okay, so during 5th week, as you're making the sex cords, some epithelial cells on the surface are detaching, going into the gonad and waiting for something. Okay, what do you think? When is the next step? This is the 5th week. When is the next step? I hear pretty much all the time 6th week. Okay, so not really. Okay, not even 6th, not even 7th, not even 8th. The next step is at puberty. We do nothing to the sex cords, absolutely nothing until puberty. The boy is born, everything is great. Only when you're hitting the puberty, there will be some kind of changes in the testes. I think it's understandable, right? So this gonad after a birth will be called testis, right? So it's one testicle. So first of all, when do you hit the puberty as a boy? It depends, like you cannot say when exactly, but approximately from 12 to 16. Okay, so this is typically the time, 12 to 16 years old. That's when, of course, there are early puberty hitters, there are late ones, but typically it's never later than 16 and never later earlier than 12. So that's the approximate time for hitting the puberty. I really like that in English we say hitting the puberty because it really shows that it is a very sudden moment, a very sudden process. Over a moment of like one night, you have overflow with testosterone in case of men and estrogen in case of female. And in boys, puberty is more visible in one moment. How? They go to sleep as boys, they wake up as men, if you know what I mean. Okay, so they wake up with the erection. So testosterone causes the erection to appear. So this is when you know that definitely the puberty is happening. The opposite is true for a female, right? So they go to sleep as a girl. Typically, you get your first menstruation at night. Okay, so you wake up as a woman. Most of the time, your first menstruation comes at night. There are some cases where it happens during the day, but typically it is at night. We'll talk about it next class, the whole hormonal situation, but for now, just imagine that there is a puberty starting, yeah? Your boy is 12 years old. There's overflow of the testosterone. There's so much testosterone in the body. What does this testosterone do? It causes, actually forces your sex cords to acquire a lumen. It forces them to get a lumen. So can I call them as the sex cords? I should call them some kind of tubules, right? Because they have a lumen now. The name of these tubules, they are known as the seminiferous tubules. So sex cords at puberty become seminiferous tubules due to testosterone. So a question for you guys. Seminiferous tubules are present at birth, correct?
No.
No.
For anatomy department, that is not correct. But you know, you reach 4th year, you reach the final practical parts, theories behind. Actually, that is correct. Because we have a bit of the testosterone in embryo. And when you're born, you have a bit of a testosterone in your blood. So it forces some sex cords to become seminiferous tubules. So if we are in 4th year, that's actually correct. But we are in 1st year, so that's incorrect. Okay, so seminiferous tubules are not present at birth because we need testosterone for their appearance, and that happens at puberty. I'm very interested to see what is inside of the seminiferous tubules. So I'm going to take one, cut it in half, cross section, and draw what do you see, right? So what's inside of one seminiferous tubule? So you see that this is one seminiferous tubule here. And there's another one here, and there's another seminiferous tubule here as well, right? There's a bunch of them just sitting together. You know that these are seminiferous tubules because in their center, you can actually see the lumen, right? So this area right here, this is the lumen of the seminiferous tubule. There is a new population of cells in the seminiferous tubule They are triangular in shape and they have 3 names. Okay, very convenient, triangular in shape, and they have 3 names. The first name for them is that they're called supporting cells. The second name, Sertoli cell. That's the one which is going to be used in embryo self-control test. Okay, so Sertoli cell. So these triangular cells of the seminiferous tubule, they're Sertoli cells. Next semester, histology, we're going to call them sustentacular cells. Second year, right there, welcomes you, all right? Sustentacular cells. 3 names, supporting, Sertoli, or sustentacular cells. What do you think? Are they PGCs? No. You remember some cells from the surface epithelium during the 5th week detached and went into the gonad and waited, you remember? So these cells from the surface epithelium have become the Sertoli cells. So the ones that during the 5th week have detached, went into the gonad, you're gonna call them the Sertoli cells. So where are my PGCs? Because last time I checked, we were having a bunch of PGCs in the gonads, right? So where are they? PGCs in embryo were sitting in the sex cord. This is seminiferous tubule. This is a different structure, right? Different name, new me. So as a result, PGCs will also change their name. PGCs are in the sex cord, but in the seminiferous tubule, PGCs are known as spermatogonia. They're the same cell. PGC and spermatogonia are identical. They're exactly the same. The only difference between them is the location. I repeat, you call them PGCs in the sex cord. The moment you're hitting puberty, the moment these sex cords become seminiferous tubules, PGCs change their name. They're becoming spermatogonia. Where my spermatogonia are sitting? They're sitting between Sertoli cells. Right between them, right here, I'm gonna have a bunch of spermatogonia just sitting there. I'm going to magnify this area, right? So this area I want to magnify because I kind of drawn it very weirdly. So I have 1 Sertoli cell here. I have another Sertoli cell here. So Sertoli cell number 1, Sertoli cell number 2. Who is sitting between 2 Sertoli cells? Who is here?
The spermatogonia.
How did you call spermatogonia before?
PGC.
And where did this PGC come from?
From the sex cord.
It is in the sex cord.
Oh.
But what is the original origin of the PGC? Where did it come from?
Epiblast,
Amazing. When?
2nd week.
2nd week. So I'm just trying to make sure that you're tracing yourself a little bit back. Okay, so PGC come from epiblast, spermatogonia come from PGCs. So what are these spermatogonia are going to be doing? Well, they have one job, is to create spermatozoa. So let's follow the steps. How do we make spermatozoas? Spermatogonia divides and gives rise to primary spermatocyte. Primary spermatocyte divides and gives rise to secondary spermatocyte. Secondary spermatocyte divides, gives rise to spermatid. Spermatid does not divide. It simply matures to become spermatozoa. So I repeat, spermatogonia divides to become primary, divides to become secondary spermatocyte. Secondary spermatocyte divides to become spermatid. Spermatid does not divide. It simply matures to become spermatozoa. So is it correct to say that spermatids are immature spermatozoa? Hell yeah. Okay, you're absolutely correct to say that. These spermatids, the precursors of spermatozoa, they are immature. Think about it this way. Spermatozoa is the adult, spermatid is the baby. Okay, so immature, mature. So 5 stages of spermatogenesis. Spermatogonia, primary, secondary, spermatid, and eventually spermatozoa. Is that clear? Where does spermatogenesis happen? As you can see, it happens between two Sertoli cells. What was the first name of the Sertoli cell? We called it supporting cell. So do you get why is it called supporting? Because it basically supports spermatogenesis, supports it nutritionally, physically, emotionally, whatever you can come up with, okay, it does it. So these supporting cells, they're just holding all of the spermatogenesis. There is a third cell. We said that there is Sertoli cell. We said that there is spermatogonia. But between seminiferous tubules, between them, there is a third type of cell. And this third type of cell, which is sitting between the seminiferous tubules, this is known as the Leydig cell. Leydig cell is the first one to appear. I repeat, we have Sertoli, we have spermatogonia. Leydig is the first one to appear between the seminiferous tubules. What is the job of the Leydig cells? Leydig cells secrete testosterone. So we have them even during embryological development. We have a bunch of Leydig cells in embryo, okay? And they're actively secreting testosterone. What is the origin of the Leydig cell? What do you think? Where did Leydig cell come from? Where did Sertoli cell come from? Meridian lip, so surface epithelium. Okay, so it comes from the surface epithelium of the gonads. Where did spermatogonia come from?
PGC.
So what is the one thing we haven't used yet?
The mesenchyme?
Mesenchyme. Leydig cells come from mesenchyme. They arise from the mesenchymal stem cells, okay, of the gonad. So Leydig cells come from that. Now, spermatozoa, is it haploid or diploid?
Haploid.
Haploid. What about spermatogonia? Is it haploid or diploid? Where did spermatogonia come from?
The PGC, so it's diploid?
So it's diploid. PGC is diploid. Spermatogonia is also going to be diploid. So now what we have to do, we have to ask ourselves, in which part do we change to become haploid? So you need to remember that spermatogonia are diploid. Primary spermatocytes are also diploid. But starting from the secondary spermatocytes down, the cells are haploid. So if I go back, spermatogonia are diploid, diploid, haploid, haploid, haploid. So primary spermatocyte is diploid, secondary is going to be haploid. As a result, between the primary and secondary spermatocyte, there is going to be a junction. There's going to be a barrier. And we call this barrier blood-testis barrier or Sertoli cell barrier. Think about it this way. Blood-testis barrier is basically a tight junction between two Sertoli cells. You can imagine that they're holding hands here. Okay, so these are the two Sertoli cells. They have a tight junction right here. Which cells are above that tight junction? Spermatogonia and primary spermatocyte. Which cells are below it? Secondary spermatocyte, spermatid, and spermatozoa. To simplify, diploid are below, haploid are above. This is why this barrier is technically not a barrier. They don't like to use the word barrier because like blood-air barrier is like 4 layers. Blood-placenta barrier is like 8 layers. Blood-thymus layer, 6 layers. Blood-brain barrier, 3 layers. Do you see what I mean? Like it's a bunch of layers. blood-testis barrier is not a barrier, it's just one tight junction. That's just one layer. So it's not technically a barrier. So that's why they don't like to use the word barrier. But still, who does this blood-testis barrier protect? Who is it hiding? Diploid or haploid cells? We said that above it are diploid, below it are haploid. So which ones are we hiding? I heard diploid, but it's actually wrong. Okay, it's haploid. We're hiding the haploid cells. From who? Who are we hiding from? So these are your Sertoli cells. They're sitting on the basement membrane. Under the basement membrane, we have connective tissue. Within this connective tissue, we have a bunch of blood vessels. The problem is that immune cells, lymphocytes, they can come here. And if lymphocytes see haploid cells, they think to themselves, what the hell is wrong with you? Like, why are you haploid? Why do you only have one set of chromosomes? This is not normal, right? You should have two sets of chromosomes. You should be diploid. So that's the logic of the lymphocyte. So what do? They want to kill them. They want to kill the haploid cells. So do you see what we have done here? We simply put a tight junction between the Sertoli cells and hid the haploid cells under that tight junction. So I have a lymphocyte coming from a blood vessel. What does it see? It sees spermatogonia. It sees primary spermatocyte. They're diploid, like the rest of the cells in my body. Everything is great, no problem, right? So we're trying to protect the haploid cells from the immune cells and occasionally virus. Okay, virus also travels in the blood. So you're gonna see that it can sometimes infect the haploid cells and not the diploid cells. So haploid cells are the ones that need protection. Is that clear? There is additional process which is called spermiogenesis. So this entire thing is called spermatogenesis. From the spermatogonia all the way to the spermatozoa, we're gonna call it spermatogenesis. Spermiogenesis is the last step. Okay, so spermiogenesis is the last step when spermatid matures to become spermatozoa. Okay, you can see all of the changes here, but let's talk about them on this picture. Get familiar with this picture, or if they put a lecture, like whatever picture we have on the lecture, because for sure they're going to bring it up. Okay, they're going to bring a picture of spermatozoa and point and ask you which area is what. So first of all, spermatozoa is 70 micrometers long, but as always, you need to double check after me. You need to go to the lectures and check the number that they have put for you. Last year, it was 60. Okay, I'm not sure this year. Okay, so last year, the length of the spermatozoa was 60 micrometers. Spermatozoa contains head, neck, okay, neck, middle piece, and tail. Tail is, of course, flagellum. Hopefully you remember from our first histo self-control test that flagellum is a super, super long cilium, right? It's a very long motile cilium. What's its arrangement? Which kind of arrangement can you find in the flagellum? If it is a cilia, so which kind of arrangement can you find? Oh, breaking my heart. You find 9 plus 2. 9 plus 2 axonymal arrangement. Do you remember this? 9 doublets and 2 singlets in the center, right? Microtubules. So this was the axonymal arrangement right there. The middle piece is very interesting in spermatozoa because it contains a bunch of mitochondria. As a result, you can think of the middle piece of spermatozoa as the little tiny engine. Okay, this is the engine of spermatozoa. It keeps on producing ATP, and this ATP is being used by the flagellum so it can move So your spermatozoa can actually kind of go places, right? Surprisingly, mitochondria are not present in the head. You don't see a single mitochondria in the head. As a result, mitochondrial DNA is always of maternal inheritance. We have mitochondrial DNA of a mom because oocytes do have mitochondria. The head of spermatozoa does not have. Okay, so this is why you can't get it into the oocyte. In the head, we have a region called acrosome. In this picture, it's kind of wrongly visualized, but acrosome actually covers 3/4 of the head. Okay, so it covers 3/4 of the head. Acrosome covers the nucleus. It kind of hides the nucleus. It's like a protective cap that makes sure that the nucleus is protected. But acrosome also has another function because it also contains digestive enzymes. And these digestive enzymes are needed to penetrate the wall of the oocyte. You know that fertilization is the fusion of the spermatozoa and the oocyte. So how can spermatozoa get into the oocyte? Only if we bulge through the wall. So to do that, we need the digestive enzymes, and these enzymes are present in the acrosome itself. Spermatozoas can survive in the female reproductive tract for 3 to 5 days, very important. The entire spermatogenesis from spermatogonia to spermatozoa takes approximately 74 days. Okay, so it takes around 74 days, the whole process. And the process of converting diploid primary spermatocyte into haploid secondary is the longest. It takes approximately 22 days. Okay, so your primary spermatocyte is kind of present in the secondary. It's present in the prophase to become secondary spermatocyte for approximately 22 days. You know that this is meiosis, right? So we have meiosis 1 and we have meiosis 2. Can somebody quickly tell me what's the difference? In a simple English, without using genetics. What's the difference between meiosis 1 and 2?
The meiosis one, sorry, I'm getting confused. Meiosis one is similar to mitosis.
Okay.
Yeah. Well, meiosis two isn't. Yeah.
Okay, so meiosis 1 in simple English is actually the division of sex cells. So this is the part where we convert our diploid cells to haploid. Okay, so meiosis 1, that's what we do. Okay, so we convert our diploid sex cells to haploid. So it's production of sex cells. Who enters meiosis 2? Secondary oocyte. Can secondary oocyte finish meiosis 2? What is the product of meiosis 2? Haploid or diploid?
Haploid.
Diploid. So can secondary oocyte finish meiosis 2? It absolutely cannot, because secondary oocyte is a haploid cell. How can one haploid cell give me diploid cell? So who needs to join the party for the secondary oocyte to finish meiosis 2?
Spermatozoa.
Spermatozoa. So what am I describing?
Fertilization.
Fertilization. So in simple English, especially next semester, it will be very useful for you guys. The simpler you speak, the higher your grade goes, because it shows that you didn't just memorize words out of the book or lectures, you actually understand what you're talking about. In simple English, meiosis 1 is the division of sex cells. That's it. So we just divide them, we produce sex cells. Meiosis 2 is fertilization, okay? Secondary oocyte cannot finish meiosis 2. It can't. It enters it, don't get me wrong, it enters it, but it cannot finish it. It needs spermatozoa because the product of meiosis 2 is zygote, fertilized oocyte. Okay, that's the product of the meiosis 2. Okay, last thing here, I want to talk about something called semen. Okay, so semen, as you know, this is the fluid the male ejaculates during, not during, after the sexual intercourse, right? So that's the fluid that comes out of the pee-pee. So the big question is, what does the semen consist of? Well, for sure, you can understand that it has spermatozoa. That's the most important thing, right? So it has spermatozoas. These spermatozoas are floating in a fluid. This fluid is known as the seminal plasma. Okay, seminal plasma. So semen is two things together: spermatozoas plus seminal plasma. Who produces spermatozoas? Seminiferous tubules. I think we established that, right? So there's a whole process happening in the seminiferous tubules to make spermatozoa. Who has seminiferous tubules? Testes, of course. Your testicles, they're filled with the seminiferous tubules. Who makes seminal plasma? The most important gland in men, which is called prostate. Do you guys know where is prostate? Where is prostate? It is under the bladder. Okay, so it's right under the bladder in men, and it kind of wraps around the urethra. So that's why with age, men have difficulties with urinating, right? Because this prostate will be constricting the urethra and not allowing the urine to pass through. So seminal plasma is produced by the prostate, okay, spermatozoa by seminiferous tubules. The question you're going to expect is this: what are the contents of seminal plasma? What is inside of it? Most important thing, fructose, because for sure you know this, that spermatozoas are unique. They don't like glucose, they like fructose. So seminal plasma is filled with fructose. It has vitamin C. This is why in some countries, semen is used as a mask for a face to kind of make your face brighter. It possesses alkylase. What's alkylase going to do?
Keep the pH high.
Yes. And why do we need it? Why do we want to keep pH high?
To neutralize the acidic environment of the
female, exactly, because you know that vagina and uterus are quite acidic. The pH is about 5.5. Okay, so it's quite acidic. So we need alkylase to make sure that we can neutralize or buffer the acidity of the female reproductive tract. So we can basically survive, to make sure that acid does not just freaking get rid of the spermatozoa. And the last thing that they want from you is this: prostaglandins. You know, I really don't like the theory years, like the first 3 years in the medical school, because it's very difficult to talk about it. Like it's, there's so many things, but they just kind of simplify it to very little things. So they just say prostaglandins. The amount of prostaglandins that we have in our body is insane. There are more than 300 of them. So it is incorrect to say that prostaglandins do just that part. Do you know what I mean? There are more than 300 of them. They have so many jobs. So this is why I myself have actually won a point arguing back in my years when I was first year, okay, that I was like, this is incorrectly phrased question. You cannot do that. But without going too much details into that, prostaglandins, the correct answer, prostaglandins cause smooth muscle contraction. For the second year, when you come to physiology, you're going to see that this statement is wrong. It's a wrong statement. Only one type of prostaglandin causes smooth muscle contraction. But for anatomy department, they couldn't care less. So this statement is correct. Prostaglandin causes smooth muscle contraction. And guess who has lots and lots of smooth muscle? Well, uterus and vagina. So basically the semen is actually helping the uterus to contract. And as a result, the uterus helps semen in return to come closer to the oocyte. This smooth muscle contraction is needed to push the semen closer to the oocyte. Where the oocytes are going to be waiting, they're going to be waiting in the fallopian tubes. Okay, so that's where they wait for the semen. Okay, I hope it wasn't difficult, because now we actually start the difficult part. We start the female. Okay, so let's talk about the female. So the idea of a female is actually quite the same. Basically, we have the PGCs. These PGCs are now carrying XX chromosome, right? So they are different. We're talking about a different human here. We have a gonad in the female as well. The gonads are the future ovaries. The same idea that these gonads have simple cuboidal epithelium on the surface and mesenchyme in the core. We discovered that PGCs carrying XX also come into the gonad during the 5th week, also divide mitotically, also align into sex cords. But what am I missing?
Y chromosomes.
Exactly. If there is no Y chromosome, there is no TDF. There is no testis determining factor. Without the TDF, the sex cords dissociate. They become clusters. So you see that there is one cluster of PGCs here. There is another cluster of PGCs here. There is another cluster here, right? So in female, the PGCs do not stay in the sex cords. They become clusters. These clusters will be surrounded by cells. These cells are called follicular cells. And where did these follicular cells come from? They came from the surface epithelium. So simultaneously as you're forming the clusters, some cells from the surface epithelium of the gonad detach, migrate into the gonad, and start to surround the clusters. Okay, so outside of the clusters of PGCs, you have follicular cells. Could you tell me, follicular cell, what is it analogous to? Which cell in male follicular cell is analogous to?
The Sertoli cells.
The Sertoli cell, because they both come from surface epithelium. The only difference between them is that Sertoli is in the man and follicular cell is in the woman. That's it. That's the only difference. But both of them come from surface epithelium of the gonads, both. So what happens next? In men, we waited until the puberty to make spermatozoa. In female, we're already late. So 5th week, this PGC will be dividing to become oogonia. Oogonia will be dividing to become primary oocyte. Primary oocyte will divide to become secondary oocyte. This is the process of oogenesis. We said spermatogenesis is 5 stages, right? Spermatogonia, primary, secondary spermatocyte, spermatid, spermatozoa. In the female, oogenesis is just 3. Oogonia, primary oocyte, secondary oocyte. And actually, this last step, when primary oocyte becomes secondary, happens only during puberty. So in embryo, you will never, ever, ever see secondary oocytes. You may see primary, but you will never see secondary oocytes in embryo because primary becoming secondary happens at puberty. So I want to do something with you guys right now, but before I do it, I just want to remind you so you get it, right? We have 2 ovaries in embryo, 2. These ovaries aren't called ovaries, they're called gonads because it's embryo. On the surface of these gonads, we have surface epithelium. In the gonads, we have clusters of PGCs. There are so many clusters. I can't possibly count how many, but there are so many clusters. Which cells surround the clusters?
Follicle cells.
Follicular cells. And these follicular cells come from
Epithelial cells.
surface, yes, surface epithelium of the gonad. Is this clear? Which week is this? This is 5th week. So what I want to do right now, I want to take just one cluster, just one, and follow the changes in that cluster throughout time. Okay, just want to look at one cluster and what are the changes in that cluster throughout time. So I'm going to draw it here. We have 5th week. In that cluster, we have only PGCs. Let's imagine there are 4, but there could be many. There could be 20, 200, doesn't matter. So this is one cluster. Is it clear? In that cluster, I have 4 PGCs. Which cells surround this cluster like this very nicely? Which cells surround clusters?
Follicular cells.
Follicular cells, thank you. So I repeat because I can see the confused faces. This is one ovary, the whole ovary. It has lots of clusters, lots. In my picture, it has 5 clusters. In real life, it could have 500, okay? It doesn't matter. What you see here is representation of one cluster only. So what happens? During the 5th week, within this cluster, you can only see PGCs. That's it. 3 months later, when your embryo reaches 4 months of age, your embryo is 4 months old, you're going to see that some PGCs have become oogonia. They have done the first step. They are now oogonia. Some PGCs are really like on fire. They have already become oogonia, and these oogonia are now becoming primary oocyte. And you see that other PGCs are procrastinating. They do nothing. They still stay as PGCs. So you look at the same cluster at 4 months age embryo. What are the changes? Do you only see PGCs in this cluster? No. What else can you see? You can also see
Oogonia.
oogonia, and you can also see
Primary oocyte.
primary oocyte. So it shows you that every cell divides at its own rate, right? They aren't doing it simultaneously. Every cell does whatever the hell it wants to do. It wants to divide, it divides. It wants to not to divide, it doesn't divide. Why I do not see secondary oocytes in this cluster?
You need to wait until puberty.
Amazing, because you need to wait until the puberty. What happens to the same cluster 1 month later? I'm reaching 5th month of the development. If you were a primary oocyte, you do nothing. You stay as the primary oocyte. If you were oogonia, you do 1 division, you become primary oocyte. But if you were PGC, you accelerate your shit, I apologize, and you become primary oocyte. Okay, you keep on accelerating the division to make sure that you're a primary oocyte. So what do you see? During 5th month of development, in the same cluster, you already see only primary oocyte. Is that clear? So again, in one cluster during 5th week, which cells are visible?
PGC.
PGCs only. During 5th week, you can only see PGCs. During 5th month, in the same cluster, you can see
Primary oocytes.
primary oocytes only. During 4th month, you can see all of them except for the secondary oocytes. Is that clear? If I were to count how many primary oocytes do I have in the ovaries during 5-month-old embryo, right? I have 2 ovaries. How many primary oocytes do I have? I would count 7 million primary oocytes. They're going to be 3.5 in one ovary, 3.5 in another ovary. And I really wanted to sink in your brain, guys. 7 million primary oocytes. All this primary oocyte has to do, it has to become secondary at puberty. Secondary oocyte will get fertilized by spermatozoa. You have a human being. So hypothetically, this primary oocyte is technically half of the human. So imagine I have a female, and in its ovaries, I have 7 million potential human beings. Hungary is 10 million, guys. I think Israel as well. Okay, more or less. Okay, so yeah. Is it too much or is it not too much? What do you think? One female, 7 million human beings in her ovaries, half of the human beings in her ovaries. Is it good for the earth? It's definitely not good for the earth. So this is why we need to do apoptosis. We have made too many humans in the ovaries. Again, when I say human, you know what I mean, right? It's half of the human. It's just the oocyte. From 5th month until 7th month, you're going to see that there will be lots and lots of apoptosis. This is one ovary right here. In this cluster, I had 100 primary oocytes, for example. In this cluster, I had 100 primary oocytes. 99 will die. In this cluster, I had 200 primary oocytes. 199 will die. In our cluster, we have 1, 2, 3, 4 primary oocytes. How many will die?
3.
3. So leaving 1 behind. So as you can see, it doesn't matter the amount of oocytes you have in the cluster. It doesn't matter. So in ovaries, I have a bunch of clusters. I have lots of clusters. And in each of this cluster, I have all the different number of oocytes, right? I could have 300, 200, 100, doesn't matter. In each of this cluster, only one of the oocyte will persist. So can I call it a cluster? Of course not. Now out of this cluster, we have in our cluster 4, I can only have 1 primary oocyte left behind. But don't forget which cell is surrounding this cell.
Follicular cells.
Follicular cells. Follicular cell is sitting right there surrounding your primary oocyte. How do we call this structure? We call it follicle. In simple English, egg. You know, we say there are eggs in ovaries, right? So you know that there are follicles in ovaries. How many follicles are there when you're born? 600 to 800,000. Okay, so you had 7 million when you were 5 months old embryo, but when you are born, out of 7 million, you actually have 600 to 800,000 primary oocytes in the follicles. Is that clear? So you made your follicles during 7th month of embryological development. Are you going to be using them anytime soon? When are you going to be using these eggs, these follicles? Hopefully at puberty, but even then it's still too early. Okay, so hopefully some other time. So we need to freeze these follicles. We're going to freeze them until later use. All right, so how are we going to do so? During 7th month of embryological development, you're going to see that this primary oocyte enters meiosis 1, meaning that primary oocyte enters meiosis 1 to become secondary oocyte. However, in the first phase of the meiosis 1, primary oocyte is arrested. It doesn't move. through the stages. It doesn't divide to become secondary oocyte. So what arrests your primary oocyte in the prophase of the meiosis one? What arrests it is the follicular cell that surrounds it. Follicular cell that surrounds the primary oocyte secretes a substance, and the substance is called OMI. OMI stands for oocyte maturation inhibitor. In the presence of oocyte maturation inhibitor, primary oocyte stays in the prophase of meiosis 1, and it doesn't divide to become secondary oocyte. So this is how we keep our primary oocytes frozen, right? We keep them in the meiosis 1 to make sure that they don't divide secondary oocyte. I repeat, who keeps them frozen, who keeps them resting? Follicular cell by secreting the OMI. Agree with me that this prophase is going to be super long. I mean, I do not know when is it going to stop. So this prolonged prophase has a name. It is known as the diplotin stage. Diplotin stage is the way of kind of arresting your oocytes, making sure that they all not become secondary. Okay, I need to do this. I apologize because I need to make sure that you understand. We have two gonads. Somebody tell me so my voice can rest. Who goes into these gonads?
PGF.
PGCs.
Oh, sorry.
Yeah, and PGCs come here when?
5th week.
Fifth week. And what do they do?
Assemble into cords.
First, they divide mitotically, assemble into cords. Cords ain't working. They become
Clusters.
clusters. How many clusters there are in both ovaries?
A lot.
600.
Very good. A lot. Okay, so that's the answer. Okay, a lot. I do not know, but I can see that Kenry is actually saying something which is correct. You're right. It's actually 600 to 800,000 of clusters, because these are follicles, right? And follicle is the remnant of the cluster. So technically you had 600 to 800, but it doesn't matter. A lot is good. Okay, so a lot of clusters. Which cells are in the clusters during 5th week?
PGC.
PGCs only. Which cells are in the clusters during 5th month?
Primary.
Primary oocyte only. How many exactly?
Billions. Oh, 7 million.
7 million. Yes, 7 million primary oocytes. Is it a lot?
Yes.
So, apoptosis. How many do we kill? Like, just give me the whole idea. How many do we kill?
1 per cluster survives.
Very good. So majority, 99% will die, right? So we're gonna just 1% of the cluster will survive. Doesn't matter the amount of the primary oocytes in the cluster, as long as only one of them survives. I cannot call it a cluster because there's only one primary oocyte in it. So what do I call it?
A follicle.
Why follicle? Why follicle?
Surrounded by follicular cells.
Perfect. Because this primary oocyte is not sitting alone, because it is surrounded by the follicular cells. What is the job of these follicular cells that surrounds the primary oocyte?
To secrete the OMI and arrest it in prophase.
And what is the job of the OMI? OMI is going to be arresting your primary oocyte in the meiosis 1, right? But why? Why do we want to arrest it in it?
So that you can use it when you get fertilized.
So you can use it when you get to puberty, right? So you can use it when you get to puberty. Yes. So we are basically trying to save the cells. We're trying to make sure that they ain't dying. Unfortunately, as you can see, you're born with 600 to 800,000 primary oocytes in the follicles. So in a way, you're born with 600 to 800,000 follicles. But you're starting the puberty with 40,000 follicles. You're born with 600, starting the puberty with 40,000. So even though you're trying to save the follicles from dying, they're still dying. Do you get it? They're still dying. There are lots of death here. So that's the big question. How come? How come they're still dying? Follicular cell is a very random cell. It starts the secretion of OMI randomly. As randomly as it starts, as randomly it can finish making OMI. So there is no control on it. Follicular cell can secrete OMI for 40 years or for 40 hours. It's a completely random process. So think about it this way. You're a child. Suddenly, follicular cell stops making OMI. What does it mean for you? If there is no OMI, what is primary oocyte wants to do?
It wants to go through with the meiosis.
Exactly. It wants to go through with the meiosis one to become secondary oocyte. But what do I need to become secondary oocyte? I need estrogen. I need estrogen. Estrogen is the major catalyzer for this reaction. It makes sure that the primary oocyte becomes secondary oocyte. But I'm a child. Do I have estrogen? Not really. So now you're left with the primary oocyte, which is awoke by the follicular cell, right? So it's not secreting OMI. And this primary oocyte cannot become secondary oocyte because you're a child. So what is it going to do? Apoptosis. So do you understand why there is such a reduction? Do you understand, right? So when you're born, 600,000 follicles. When you start the puberty, 40,000. Whose fault is this? Follicular cells, okay? So they aren't doing their job properly. And I really, every time I teach this part, you know, I'm not a feminist whatsoever. Like I don't actually, you know, believe in that whatsoever, but this part actually makes me want to cry because we started with 7 million freaking primary oocytes. And when you start the puberty, it's 40,000. But luckily, this number is only for anatomy department. In real life, it's more like 240,000. Okay, so as a doctor, you should know that you start the puberty with 240,000. Only for anatomy department, only for the self-control test, you need to remember it's 40,000. Okay, guys, you start the puberty with 40,000 follicles. How many do you choose monthly?
It's 2, one from each ovary.
Actually, it's 15 to 20.
Really?
Every month, out of this 40,000, we choose 15 to 20 because, you know, we're girls, we need options, lots of choices, lots of stuff like that. So 15 to 20 follicles. How many of them will mature? How many do you ovulate?
1.
Just 1. So I choose 15 and only one of them is going to be ovulated. What's going to happen to the 14 of them?
Apoptosis.
Apoptosis. I forever associate the word apoptosis with a female because of this. Okay, so because you can see how much cell death there is happening. Like oocytes are dying left and right. How do you call it when you're done, when there aren't any more follicles anymore? For sure you know it.
Menopause.
Menopause, yeah. Okay, so that's when there is no menstruation anymore. That's it, there are no more follicles. So if actually anatomy department number was true, when would you reach menopause? Taking 15 to 20 follicles every month. You would reach like 28, 30 years old, okay? So that's when you will reach the menopause. But that's not the case, you know, you reach closer to 40, 50 years of age. So this is why it's more like 240,000, okay? So that's the correct number. Okay, guys, we aren't even close to done with the female. We continue. So a little bit, I'm just gonna introduce this slide, and this slide is already next class. Let's start puberty in the female. Okay, so puberty in the female starts with hypothalamus. Hypothalamus is a very important gland in the brain. It controls lots of processes. One of them is just reproductive cycles. Hypothalamus secretes a hormone which is known as the GnRH, or gonadotropin releasing hormone. This means nothing to you. We're going to learn more about it next semester, but unfortunately they ask about it this semester. Okay, so hypothalamus secretes this hormone, gonadotropin releasing hormone. This hormone acts on the anterior pituitary gland. It's also a gland near our brain. It's sitting in the sella turcica. I'm not sure if you know what is that. Turkish saddle, pituitary fossa, skull, right? So that's where the pituitary gland is sitting. And this GnRH is stimulating anterior pituitary gland to secrete gonadotropins, because think about the name of this hormone, gonadotropin releasing hormone. It is asking you to release gonadotropins. So which hormones are gonadotropins? This is going to be your FSH and LH. These are the two gonadotropins. FSH stands for follicle stimulating hormone. Follicle stimulating hormone. LH stands for luteinizing hormone. Which one is important for growth of follicles? What do you think?
FSH.
For sure, FSH, follicle stimulating hormone. Okay, we said that we had 2 ovaries, right? Hopefully. Each ovary hopefully had 20K of follicles, am I right? Total number is 40K. How many follicles do we choose every month out of this 40K?
15 to 20.
15 to 20. How do we choose them? How do we make sure that these 15 to 20 are the chosen ones? I volunteer, you know, one of the follicles. I volunteer as a tribute, you know. So yeah, but no, that's not the case. Basically, the FSH. Okay, FSH is partially responsible for that. So again, there is going to be a random process every month, randomly, 15 to 20 follicles out of the 40,000 raise their hand, literally, which is the receptor. Okay, so randomly they express receptor for FSH. The key word for the female is random. Everything here happens randomly. Okay, so randomly every month, out of the 40,000, 15 to 20 follicles raise their hand, which is the receptor for FSH. So when FSH is being secreted, when it comes to the ovaries, it doesn't stimulate all of the follicles to grow. It only stimulates the chosen ones, the ones that have the receptor for the FSH. So let's take a look. The follicles that we have made when we were an embryo are called primordial follicles, because primordial means before birth. So those 600 to 800,000 of primordial follicles, those 40,000 of primordial follicle. So how does a primordial follicle look like? So of course, inside of the primordial follicle, you're going to see primary oocyte. Nothing have changed. So this is the nucleus of the primary oocyte. This is the cytoplasm of the primary oocyte. Which cell surrounds this? Of course, the follicular cells. The shape of the follicular cell in the primordial follicle is flat. It is squamous. Somewhere here, there's going to be a hand, a receptor for FSH. So when FSH comes in, it binds to this primordial follicle and forces it to grow. It asks it to be stimulated, to grow, to go through some kind of stages. Under the influence of the FSH, primordial follicle becomes primary. This is a mistake in the book. This is also primary. It's just that we see two stages of the growth of the primary follicle. This is the primary, this is the first stage of the primary follicle. This is the second stage of the primary follicle. What is the difference between primordial and primary follicle? Inside, no changes. You still see primary oocyte. Nucleus, cytoplasm of the primary oocyte, 0 changes. What has changed? The shape of the follicular cells. They were simple squamous in the primordial, and they're simple cuboidal in the first stage of the primary follicle. Still, there is a hand, still there is a receptor for FSH. So FSH comes in, binds to this follicle, forces it to grow more. So we see that the first stage of the primary follicle becomes the second. So what are the changes? Again, inside, no changes. Still, it is a primary oocyte inside. But you can see that the follicular cells are no longer simple cuboidal. You see that there are several layers of them here. So they're actually stratified cuboidal follicular cells. So I repeat, main difference between the first stage and the second stage of the primary follicle is what is the epithelium of the follicular cells, right? In the first stage, it is simple cuboidal. In the second stage, it is stratified cuboidal follicular cells. These stratified cuboidal follicular cells look like little granules. They look like little tiny dots. As a result, they are also known as the granulosa cells. Okay, you're going to call them as the granulosa cells. So a question for you: at which follicle stage granulosa appear? At primary follicle second stage. That's when they appear. What are granulosa cells? Stratified cuboidal follicular cells. What is the function of granulosa cells? They secrete glycoprotein coat. This glycoprotein is coating your primary oocyte. This glycoprotein is known as zona pellucida. Do you remember spermatozoa had acrosome, right? And acrosome had digestive enzymes. And we said we needed these digestive enzymes to penetrate a wall of the oocyte. Which wall? Zona pellucida. We need to penetrate this wall because it's a glycoprotein, like a sugary wall that is kind of outside of the primary oocyte. So again, what are the layers of the primary follicle? Inner is the primary oocyte, then zona pellucida, then granulosa cells. Is it clear? I go back. This is a piece of ovary. One little cuboidal piece of ovary. This is one primary follicle. This is another primordial follicle, doesn't matter. This is another primary follicle. What is this tissue? This white. This is connective tissue of the ovary. Connective tissue of the ovary, which directly surrounds the follicle, has a name. It is called theca folliculi. Okay, theca folliculi. So what is outside of my primordial follicle? What is this? This is connective tissue of the ovary, which is called theca folliculi. The same here. Do you see? This is the connective tissue of the ovary. It doesn't have a very particular, I don't know, different composition. It's the same connective tissue. It's just that they had to have a name for it. Okay. So is this theca folliculi? No, because it isn't next to the follicle. Is this theca folliculi? this is your follicle. Is this theca folliculi?
Yeah.
Okay, it's the connective tissue which directly surrounds the follicle. So yeah, that's theca folliculi. Do you get it? So just an empty connective tissue, not next to the follicle, no name. As soon as it is next to the follicle, theca folliculi. Okay, last thing. I promise, last thing, last sentence, literally. Primary follicle should become secondary. Still, there's going to be a receptor for FSH. FSH comes in, stimulates this follicle to grow, right? So this follicle can become secondary follicle. Unfortunately, I don't have a picture for it here. I promise next class, tomorrow, or whenever I'm meeting you, I'm gonna give you the picture. But basically, the major difference between primary follicle and secondary follicle is a little tiny space. Okay, there is a space here, little tiny cavity. If you see this cavity surrounded by granulosa cells, this is a secondary follicle. If you don't see this little cavity, this is a primary follicle. Because the next stage is the tertiary follicle stage. And notice how big the cavity becomes, right? It is a huge cavity. So that's the major difference. Secondary follicle has a super tiny, little tiny cavity in the granulosa cells layer. Tertiary follicle has a huge one. Last sentence. How many follicles do we choose every month?
15 to 20.
How many of them will reach the tertiary follicle stage? As you can understand, tertiary follicle stage is the final stage. So how many of them will reach this stage of development?
1.
Just one. So what will happen to the rest of them? Apoptosis. Can it be 2? Can we have 2 tertiary follicles? What will happen to them?
Get twins.
Yeah, if there are spermatozoa, if the female had sex during that time, definitely these 2 tertiary follicles will eventually get fertilized, right? And you're going to see that you're going to have twins. But which kind of twins? Identical or non-identical?
If
Non-identical.
it's 2,
Non-identical, because I'm going to have 2 follicles. I'm going to have 2 tertiary follicles, 2 oocytes. These 2 oocytes are going to get fertilized by 2 spermatozoa. So completely different human beings are going to arise, right? So 2 non-identical or fraternal twins. Okay, guys, so that's it for today. Thank you for surviving this 9 p.m. Okay, so we're gonna start with this slide tomorrow. This whole PowerPoint is not the only one. I'm gonna send the second part, the last one, tomorrow. Okay, so tomorrow I'm going to send it for you. Let me start the recording.